Thanks, Tina. It's a pleasure to be here, and this is Tina's last time with this course. Hopefully, we're going to continue doing courses worldwide. She was in India with me in December, and I hope that she stays involved in teaching because she's it's been incredible, and she was I think the driving force behind creating this format that you have here today, and so yeah, also the first time I ever came to Zurich, I met Tina, and I said, "Oh, you should try my software." And she was just getting started with the Aerosolve lab, and she said, "Oh, well, I'm not really a software person because a lot of times you have at these labs you have informatics people doing all the analysis. Certainly, the Aerosolve lab has several informatics people generally, but my response was no, do you use Excel? That's really what I'm trying to create is a tool for people who know how to use Excel. um, so that's as Tina said, that's sort of what I've been targeting, very user friendly software that you don't have to be a software expert to use. And that started with this interlab collaboration uh, of the National Cancer Institute's Clinical Proteomics Technology Assessment for Cancer Verification Working Group. They did a set of studies, and they paid for Skyline. The initial 3 years of Skyline was funded by this group, and they were doing these interlaboratory studies involving 12 sites, 4 instrument manufacturers, 7 different triple quadrupole instrument models. As we developed Skyline to meet those needs, it was really exciting to see the amount of sharing that was going on among these labs that hadn't been possible before. So they were sharing their mass spectrometric methods, spectral libraries, data analysis, and reports. And they pooled these reports from all of these labs, and then did shared data analysis using R. And so that was, and a very important use case was that all of these 12 labs with 7 different types of instruments could process their data in the same way, get a single report out, and then do the statistical analysis in a shared setting. And then, yeah, the goal was reproducible research, and the goal was to understand how reproducibly we could do these types of experiments, and that's now published in these 2 papers. Um, so Skyline itself has grown since then. So in those days, it started out as a tool for selected reaction monitoring (SRM), which is, so you're gonna sort of progress through the history of Skyline yourself in this course. We're starting with SRM. Um, but now Skyline supports all of the other methods that Tina mentioned, including actually doing similar experiments using DDA data, where we extract the chromatograms from the MS1 scans. Um, so when I think of these 4 methods, for which there are multiple names that Tina mentioned, I think of them in this 2 by 2 matrix, where we have targeted methods, where we're doing targeted acquisition. We're actually telling the instrument what we want to measure, versus survey acquisition, where you generally set the instrument up with a sort of broad set of settings, and then measure. You're measuring everything essentially, and you can go back and query it later. I also think about selectivity, and the more selective methods and the less selective methods will translate into your dynamic range when you do your experiments. And so PRM we're now feeling is more selective than SRM, and then DIA more selective than DDA, and that was a little bit shown in Rudy's slide, where he was showing that you got lower limits of quantification with the DIA data than with the DDA data. So the MS, when you're extracting chromatograms from fragment ions, you can get better sensitivity, more dynamic range than when you're extracting from the MS1 scans. And then this This is an image created by Jarrett. You'll probably see this again in animated mode. Jarrett has a really nice way of showing this. This is the three-dimensional space that we're always exploring in mass spectrometry, which is intensity, mass to charge ratio, and retention time. And so in this case, there's a set of scans here, and Skyline is then extracting chromatograms out of these scans, and then what you'll be seeing when you start using Skyline is a lot of plots that look like this, where all these scans have been collapsed to just a set of chromatograms in intensity and retention time. So the project, Skyline as a project, as Tina said, it's in its 7th year of development. It's almost done with its 7th year of development. Started around August 15th. There are now 6 developers, over 400,000 lines of code. It's all open source. There are 60,000 lines of code for testing. We're a group of professional developers who've worked on a lot of projects, and we're all really proud of the level of testing and the level of automated testing that we're doing with Skyline. We think that's one of its great assets, and so it is heavily tested, and that's one of the things that allows us to continue forward without breaking it. There's quite a bit there's nearly 6,000 registered users now, and then over 7,000 instances started each week. And then it's funded by NIH grants, and then all 6 instrument vendors that it supports are now helping to fund Skyline. And this is what things looked like toward a little bit earlier in this year, and then this is what it looks like now. We made another release, 3.1, and you can see that there's a lot of interest in 3.1. And then I guess I like to say that if I've learned anything quantitative over the last seven years, it's that not a lot of proteomics happens around Christmas and New Year's. So, and as Skyline now supports instruments from these 6 instrument vendors, the original 4 were Agilent, AB Sciax, Thermo, and Waters. Those were the ones funded. I was funded to support by the CPTAC program, and then Shimadzu and Bruker came along later, and they're all now helping to fund improvements on Skyline going forward. So Skyline is also very worldwide. You can use your Skyline skills to pretty much go anywhere these days, and because it's cross-instrument vendor, you can go into any lab and process data with Skyline, regardless of what type of instrument they have. So last 6 months, these are the areas in the world where Skyline's getting used. This big spot here is the not set. Those are maybe people using VPNs or something. We don't know where they're coming from. But as you can see, there's a big blue spot here. That's Seattle, and there's also one under this mass of Europe, which is Zurich. And if we zoom into Europe, we can actually see that big blue spot. Zurich is the the place where we see the most visits to the Skyline website out of everywhere, but we can see also that there's quite a lot of different places in Europe now accessing the Skyline website. We're here at a targeted proteomics course. I think a lot about why would you go targeted? I think we've had some pretty good explanations of that, but this is how I look at it. So certainly dynamic range, we get better dynamic range if we go targeted. That's been shown. Precision, cost-effective verification and validation. So compared to antibody assays, which was where the state of the art, certainly when I started, I guess Rudy was just a key author on a paper saying maybe we should flip things around and require antibody assays to be validated by mass spectrometry now. And then finally reproducibility, which Rudy did a nice job of covering. So also we have this problem in proteomics. It's an issue that sort of plagues us of doing massive multiple hypothesis testing and dealing with the error rates that entails. And Rudy showed some of the issues with that. But yeah, so with just peptide searching, it's an issue that's plagued us a little bit. And Lucas Reiter, who we're going to hear from later in the week, had a really nice paper in 2009 showing that the error rates that people were reporting at that time were probably way underreported. And this was, well after Rudy's correction that he showed, where early in, it was like 2003 or something, what was it? Yeah, 2005 or something like that. Error rates really started coming down, or the number of peptides being reported started coming down as we applied better statistics. Well, we found in 2009, and I was at a lab right around that time, a little earlier, where I definitely felt like that this was happening, where the number of proteins being reported was, they probably had like a 30 to 40% false discovery rate when they thought they had a 5% false discovery rate, and that was just because we had this tendency to do false discovery rate cutoffs based on peptide spectrum matches and then roll up all the way to proteins, and at each step along that way, error rates got inflated. So we were not doing a good job of reporting error rates. Then earlier this year, Rudy also mentioned this. We had two big papers saying that they had measured the entire human proteome, and then very quickly followed by people saying that the error rates were overinflated, and they'd found peptides on the Y chromosome that were in the ovary tissue and stuff like that. So even for peptide spectrum matching, peptide identification, which we've been doing for 20 years, we still have this problem of when we're doing these massive multiple testing experiments, we have a hard time estimating and controlling error rates. And then there is this problem of prevalence and predictive value, which I've added a slide to kind of cover. So I'll get to that next. And I thought I would like to go. If I have DDA runs and 50,000 peptides at a 1% FDR, what is the error rate for quantification? So in general, a lot of our experiments are controlled for error rate at this peptide ID level. We're all used to using DDA, and we think that, despite issues up here, we feel like we're pretty good at controlling error rates for peptide identification, but actually coming up with quantitative values involves a new set of statistics and a new set of error rates that we have to control. And then this over here on the side, which I probably should have explained at the beginning, is this matrix of replicates by analytes. So these are peptides, it could be proteins. We'd all love to just have a tool that spat out this, that gave us this matrix. And in fact, there are certainly tools that claim to do this, but it's pretty, this is a complicated matrix to produce from mass spec data. There's a lot of work that goes on under it, and if you accept a tool that just gives you this matrix, you may be accepting a lot of errors that you don't understand and that you have a hard time looking into. And I'm going to be going into that in my following slides. So this is this idea of prevalence and predictive value. If I have a test and it's got a certain sensitivity and specificity, this is an incredibly good test, 99% sensitive and 99.9% specific. So if I have that test and I apply it to 50,000 trials, then what I end up with really depends on my prior probabilities. This is basic Bayesian statistics, that if I throw something at this test that has a lot of true positives in it and not very many negatives, then I'll do much better than if I actually throw massive amounts of negatives. So and very few positives. So this is just, if I have this test and I and I try it against 50,000 all negatives, right? I don't actually have any positives. I do 50,000 negatives. I'll still come up with 50 things. I'll come up with 50 positives because I've got this 99.9% specificity, and so I'll have an 100% false discovery rate. If I now assume, I've got 50 positives out of my 50,000 negatives, then I'm going to get a 50% false discovery rate because I'll come up with 50 false positives and 49.5 true positives. So if my prior probability gives me 500 positives, and so you go down here, and eventually you end up with a 1% FDR at around 5,000 true positives and 5,000 true negatives. And then so the test, if you have a test of fixed sensitivity and specificity, the prevalence of what you're looking for impacts what you're going to get out. Okay, so that's a really important concept, and we can use statistics to adjust for this, and we can use statistics, and maybe we'll be able to not end up with all these false positives. But in order to do that, then we're going to have to sacrifice our ability to see the negatives, or to see the positives. So on air, okay, so if we have DDA runs, 50,000 peptides at a 1% FDR, each is assigned a quantitative value from 10 runs, so we're maybe 100 runs. If we're expanding this now from IDs to 50,000 IDs to across multiple runs, this step involves a certain amount of error. And now if we end up and we say, 100 of them seem to be changing by at least twofold, and we add a p-value of less than 0.05, so we're doing a t-test, and we t-test everything, and we go, 100 of them seem to be changing. Already, if you remember this slide, 100's a really low number, right? Like 100's closer to this 50%, so maybe I should, how good is that test? That t-test has a certain sensitivity and a certain specificity. Is it this good? Especially if I'm using 0.05 as my cutoff. And then 90 of them are already explained by the literature, so I celebrate. Wow, this is definitely working because I found 90 things that are already explained by the literature. And then 10 are novel and exciting. It's novel. They're novel, so I'm excited by that. Wow, 10 of these nobody's ever found. But what now if 1,000 labs do a similar process and come up with their own 10 peptides, what's the false discovery rate among those 10,000 peptides? I would say that it's certainly a lot higher than 1%, probably higher than 5%, quite possibly as high as 90% or more. So that's why we do validation experiments. That's why targeted proteomics is so important. If you think about clinical studies, governments impose multiple phases on clinical studies to avoid just going through this once and going, aha, I now know the answer. And that's why we have targeted proteomics. So here are a couple of case studies. Case study number 1, using MS Quant tools, an unnamed postdoc, unnamed MS1 extraction software, trusted quantitative expression numbers for a year or two, ran statistics, biological conclusions, came up with a story to match the results that this person was getting out of these tools, and then finally, later, they had gone a certain way, and Skyline made it possible to do the same MS1 extraction. The import of the data into Skyline found much higher error rates than they had expected, and abandoned the effort. My lesson of this is seek high visibility. So one of the things you're going to find when you start working with Skyline is that it really tries hard to let you verify, look at the data, and make sure that the conclusions you're coming up with are borne out by your data. Case study number 2, Tagachi's a grad student at the UW Genome Sciences Department. His goal was to measure proteins regulating DNA transcription. He mostly failed using data-dependent acquisition to even identify. So he purchased a cDNA library, expressed 700 proteins that he was interested in, and then did targeted measurement of all tryptic peptides for those proteins in water. A sample matrix. So again, so far he's done all synthetics, and then finally he was able to successfully measure the analyte in matrix. So here, this is a workflow that involved no DDA, right? This is a start from targeted workflow where he knew what he wanted to target, and then he just went after those things and was able to successfully measure them in the end. But that sort of a targeted or a discovery workflow probably would not have found for him, even with modern. Well, maybe not with you might have still been able to do it with modern DIA. What we're doing with Swath and DIA, and we'll find out more about that later. This is just a quick slide to cover the idea of labeled internal standards in Skyline. Labeled internal standards are sort of the gold standard in targeted quantitative proteomics. They give you here you see the blue is a heavy isotope labeled peptide. It's the chromatogram for that, and the red is the analyte. And when they coalute, then you have very, very high confidence that you are measuring your peptide of interest. And you get much better quantitative results because you can normalize to it. And Skyline was built to handle this and handles it quite well. And you're going to see some other slides. And I wanted to orient you to usually the blue is going to be the heavy and the red is going to be the analyte. And this is what the form inside Skyline looks like. You'll be seeing, certainly in the tutorials, you'll be seeing data that has this. And here's another slide showing this is data from the CPTAC study before the one that Skyline was built to handle. So this is importing that data into it. This has some of the nicest chromatography of any data set I've seen. So if you're using Skyline, you're doing chromatography-based quantification, and how successful you are depends on how good your chromatography is. So if you have chromatography like this, Skyline is going to have a really easy time processing your data. The retention time barely budges over 40 or so runs. You have really nice smooth curves for the peaks, and then so this is the retention time plot in Skyline. This is the peak area plot in Skyline. Who can tell me what type of experiment this was? What? Well, it's got aqua peptides, so the heavy are labeled. But so what is true of the labeled here, right? They're injected at a constant concentration, and now the red is the analyte. So what's going on with the analyte? It's a dilution series, right? And so how many replicates? 4. So yeah, so you see this stair step. Just immediately you can just look at this and go, oh, it's a dilution series in quadruplicate. So and very nice. Like you can see that there's probably very, very low CV for these measurements. This is what poor chromatography looks like. When I started this, the woman who wrote MS Stats, Mina Choi, said she'd been getting data from the Aerosolid lab and data from the McCoss lab and other top mass spec laboratories. She's like, what does bad chromatography look like? So I added this slide. So these are 2-minute wide windows. There's a lot of instability in the signal, and so amazingly, the peak picking package that was written by a graduate student at the McCoss Lab, actually picks this as a single peak. So this is Skyline's default integration of this. I've seen worse chromatography, and one of my next slides is going to show some where Skyline does not succeed in integrating the whole peak. But you're still, even if Skyline is getting your peaks right like this, you're still going to get better quantitative results if you have chromatography like this versus chromatography like this. And here's the slide with even more jagged chromatography. And if you think, oh, maybe it's because it's really low abundance, you'll see that this is up at 100 million ions per second. So this is a fairly intense peak. So some sources of error that, like we would like to have a tool that would just generate, the matrix I showed you, but the truth is that frequently we're unable to because of just issues with the data and the complexity of the data. And so, in this case, the chromatography, Skyline has chopped this peak, and we would need to adjust this to get the correct quantitative value for this peptide. And then appearance is a huge problem almost always in a complex matrix. In this case, this is the way Skyline by default integrated this, and you would prefer in this case to have Skyline put the integration boundary between this and the other thing, but the fact is there's this other peak next to it is going to be a problem for measuring this, right? Because chromatography is not exact. There's already definitely some signal coming from this peptide into this third transition here, so you would want to exclude this transition. But so I guess again the sources of error here are the interference, and then the integration boundaries could be better placed, but you couldn't really get rid of the interference altogether unless you delete this brown transition. Here's just an incorrectly picked peak. This was a bug that Skyline picked this peak instead of the one with the ID in the middle of it. And if you think, oh, yeah, boy, now you may be thinking, oh, Skyline's terrible software. It's it's allowing all these sources of error. Why doesn't it just get everything right? The truth is that all the other software you use will have bugs and will not get this perfectly right. And the thing about Skyline is it gives you visibility into it, and it does give you a chance to correct it if you can figure that out. But something that would just give you the matrix to start with is hiding that all from you. And so you've just got this false sense of security that like, oh yeah, I've got quantitative values. They just gave me quantitative values. And what you really need to do is make sure that before you publish, you at least find some tool that will allow you to really closely check your results. Here's another case. There are just some peptides are just really hard to quantify. And if you were doing a label-free experiment, this would be one of them. You might still be able to get a reasonable quantitative value out of a labeled experiment with a reference peptide. But in this case, and we will probably actually look at this later in the week, a number of the replicates looked like this, which is really less than half minute. This looks just like our good chromatography example. But over here, now we're out to 2-minute wide, double eluting, and it's really hard to figure out how you would integrate this to be a comparable quantitative value to this other one on the left. So in this case, I think the best solution is to just say this is not quantifiable. Another thing that we're now checking for regularly when we do quantitative targeted proteomics, so we spend a fair amount of time getting to know the peptides that we're going to be quantifying because experience has shown us that part of targeting is understanding the peptides and picking the right peptides and continuing to refine our methods to better and better measure these things. And so one thing is that we see autosampler degradation. So some peptides just don't hold up very well, and so they degrade over time in the autosampler. So we now frequently do a 72-hour wait between measurements to make sure that we have stable peptide measurements. So this one degrades 200-fold over the course of this experiment. And if we were to take new sample fresh out of the fridge, it would pop right back up to this level. And so this, again, is something we wouldn't want to use for quantitative measurement. It turns out that we actually had this, and we're going to see this later in the week. This peptide was intended to be a global internal standard. So we thought we were going to try to normalize everything else to this. But now looking at this, we realize that's not a good idea. But ideally, we would have figured that out earlier. This is what, another thing to notice, because it's going to become important when we get to another slide, is that there's no randomization here. So this is the actual order that these samples were acquired. You can see that there are 3 cycles. So this is in triplicate. But you can see it's 102, 103, 108. It's sort of sorted numerically. And sorted in the same order in every cycle. And so when Olga Vitek is here talking about statistics, she's going to explain why this is a really bad idea. We'll get a little glimpse into it right now, or on the next slide. So this is what we actually used as the global internal standard. It does also have some degradation of signal over time, so you can see it's going down. Most peptides in this experiment decreased over time, and so this is sort of normal signal decline based on the instrumentation. So when we normalized, and then we used very standard fold change statistics, we found that our 3 peptides that we had chosen for standards, because 2 of them had this signal degradation, it showed up as having a significant change. So when we look at, this is the confidence interval, this is a 99% confidence interval, you can see it doesn't go over 0, and this is the log fold change. So this protein looked like it was changing, but it wasn't. And you can see that there's a bias in this data toward fold changes that were upregulated from healthy to diseased. And now if we go back and look at this ordering, you can see that we're measuring disease before we're measuring healthy. And so that means that if we have signal degradation like this, we'll end up with data that says, hey, diseased is more intense than healthy. So that is like, proof of, if we'd randomized, we probably wouldn't be seeing this. So there are lots of mistakes that you can make in these quantitative experiments. And so in this data set, I would be very dubious about all of the peptides right in here. These are completely bucking the trend, and these are probably big enough that they may still be valid. And then here's a problem for global normalization. This is a data set that came out of the Aebersold lab, and it's a really nice, large set of replicates. It's like 80 replicates, I think. And when I started looking at the data, I started noting them. A lot of peptides look sort of like this. This is fairly extreme. They didn't all go up this much, but they, sort of the signal went up, and at some point in these runs. And this was before I had information in Skyline about what time things were acquired. I added that ability to Skyline so that you too could now fairly easily figure out that the reason this set has much higher signal is that it was collected a month after this one. So presumably they cleaned something, or they did some maintenance on the mass spec that caused signal to go up here. But that wasn't universal. There were actually some peptides also that went down. Right? So peptides don't behave exactly the same way. And so I've labeled this as a problem for global normalization, because you can't pick any set of peptides that you would normalize by and get the right answer for both of these. And that's why having these global standards that you can, or not global standards, having reference standards where you actually have the peptide and normalizing against that works a lot better. And so if you're really concerned about quantification, you should be using reference standards. And you can see here, if we normalize everything to the reference standards, you no longer can see the difference. Okay, so wrapping up, Skyline has grown beyond. So first, it's grown beyond SRM. It now handles all the 4 different chromatography-based quantitative methods. It's also we've been working closely with a group of people, and there are more people now. We've created an interface that allows people to create external tools. These were the first 6 external tools. There are 2 more and several more on the way. And they're tools that install right into Skyline, and then you can access them off of the tools menu in Skyline. You're gonna be working with MS Stats, which is by far the most popular. These tools have been downloaded lots and lots of times. MS Stats over 4,000 times at this point, and there are more of these tools on the way that take algorithms from groups that are not the Skyline team and integrate them directly with Skyline. Also, the Skyline team has created a repository, a website database repository where you can upload Skyline results, and you can use that to publish fully annotated Skyline documents, or just store fully annotated Skyline documents for your later use. And you can also use it to build these things we call chromatogram libraries, where you're using prior targeted experiments to inform your future targeted experiments. So it's the targeted equivalent to a spectral library. And then we've just recently been doing a lot of work on, and are pretty excited about this ability to aggregate QC data. So we do QC of all of our mass spec instruments in the McCoss lab using targeted documents, importing the data into Skyline, and now we can upload that data to Panorama and then go navigate it, and it will also detect when things are out of control, and so this is a pretty exciting use of Skyline and Panorama, which are not. It's not directly for a targeted experiment. It could be used for any instrument to do quality control on it. It's free, there's a free hosted version. You all can sign up for a project or join a project if somebody in your lab has already started one. There are 90 separate projects so far, over 90, and 3 big projects that we've done with the CPTAC group, Lynx, and AVRF PRG. 1,000 data sets uploaded. User-controlled security. So once you create a project, you control who gets to access the data. So you could keep all of that completely private, or you could actually make it public and share it. And then there's also a locally installed version that a number of large pharma companies have, but otherwise it's all free and open source, just like Skyline. Another project that's part of the Skyline ecosystem is this Chorus project. It's also a website. It's for raw data storage. So Panorama is for processed data. It's data that's been processed with Skyline. This is for raw data, and I'm just going to quickly show an example of how Skyline works with Chorus. Query it. You don't have to have it on disk anymore. This is a demo of that. I did a comparison on my laptop of what it would be like to use Chorus versus on my local disk. To get the data onto my local disk, I had to download it from some server somewhere, and at the time, I think I probably was not at the University of Washington. It took a while to download this. It took me an hour and a half to download 15 gigabytes. And then a side-by-side comparison of a fairly limited analysis, only 279 transitions for 29 peptides, but we do experiments like that. And that was what it was like to do local. And then when I go to import results with Chorus, I can just click on a little Chorus button, and now I'm up. This is data in the cloud on the website. I can navigate through it, and just like it was on my disk, find the same 4 files, select them, and import them into Skyline. And then this is what the import looks like. So you can see Chorus is already starting to get some chromatograms for its peptides, and the raw data local Skyline is starting to walk through the scans in the file, extracting chromatograms. And at this point, Chorus is working on its third file, and here we are walking through scan by scan. So when you're working with these full scan files, extracting chromatograms, they're very, they're gigabytes a piece, and getting through all the scans can take a while. And Skyline walks through and extracts the chromatograms. Performance of this, but we're somewhat limited by how quickly we can get these scans from the instrument vendor's software. Whereas with Chorus, once we've uploaded data to Chorus, Chorus can pre-process the data and then give it to us very, very quickly. So here it is, all 6 files have been imported into Skyline. I can look at the chromatograms, and here I can see, I can actually click on the chromatograms and look at the underlying spectra from which the chromatograms were extracted. And now here I'm clicking on the precursors, and I can see that if I click on this bump next to it, I can figure out that that bump next to it doesn't actually come from the same peptide, because it's got an isotopic distribution that's different. And here I'm back to just still walking through the scans. So that's pretty exciting. And that's, we hope that people are storing a lot more data on Chorus and using it in that way. We've, in the last 6 months, added support for all of the instrument vendors that Skyline supports. Also, Skyline is moving to supporting small molecules. Is there anybody here interested in small molecule quantification on mass spectrometers? Oh, there's one person. Okay. Well, there are a lot of people who are interested, and a lot of people who've seen what Skyline can do for proteomics people, and they go, "Oh, I want to be able to do that too." And so we've been extending Skyline to small molecules. You can see the icons change a little bit over here. When you leave here, hopefully you're gonna know so much more about Skyline than when you arrived. But there's lots and lots of extra information on the Skyline website. There's 14 tutorials. I can only show 12 of them here. I've been doing a webinar series. Now we've got, we just did our 8th session of the webinar series. And so there are companion webinars for a lot of these tutorials. And then I'd just like to introduce the entire Skyline team here. Josh is actually part of LabKey, but he helps us with Panorama. And then otherwise, these are the people creating a lot of the features that you're gonna hopefully know and love by the end of this week. It's also taking quite a lot of collaborators. There are a lot of great people who've decided to contribute data and time and suggestions and feedback, and a lot of great collaboration that Skyline couldn't be what it is today without that. I think neither could it be what it is today without the support of the instrument vendors. From the start, they've been very, very supportive with their time and very immediately interested in Skyline and seeing it be successful, and more recently to the extent that they're actually helping to fund it. And that's the end of my presentation. Do we have time for questions? Okay, questions? Anybody? Not currently. No intention to do a Skyline app in Chorus. Yeah, you run it. Yeah, so Skyline runs locally on a client computer. There's there's years of development to make it that way. There are features that would be a lot harder to do up on a cloud. I'm sure that a company like Google could give it a pretty good shot, but we don't have the budget to do that. But yeah, so the responsiveness is hard to beat of actually having the chromatogram data on your computer. But we're working on the main bottleneck for us is getting the chromatogram data, and so now we can get access to that data that's up on the cloud in Chorus using Skyline, but give you the same sort of rich client experience with Skyline with full undo, redo, and all the things that Skyline allows. So there's not really an immediate plan to move to the cloud. Anyone else? Well, I'm here for the rest of the week, and I think you're gonna learn a lot more about Skyline. I'm sure you'll have some questions as you do.